All right. So, hello everyone. My name is Isaiah Grant. I'm currently working in the lab of Dr. Blaha in the biochemistry department, and I'll be talking about the conservation of the transcription translation coupling complex in Bacillus subtilis. So, every living organism goes through the process of transcription and translation. So, transcription being the process of taking DNA and going to RNA through RNA polymerase, and then translation being the process of taking that RNA, going through the ribosome, and coming out into protein. Something that's special about bacteria is that they can couple transcription and translation together. So that means that while our RNA polymerase is taking that DNA and turning it into RNA, a ribosome can come along on that recently transcribed RNA and start converting that RNA into protein simultaneously. And the cool thing about this is that it's actually been observed. So here we have a figure from a Miller paper, and it's kind of hard to see what we have here. So um, I'm just gonna color code some things to make it a little easier to see. So here in the pink, we have a strand of DNA, and then coming off that DNA and these long lines, that would be the RNA. And then these larger dots in the figure would be the RNA polymerase, which is actually making that RNA. And then these large circles along the RNA, those would be the ribosomes actually turning that RNA into protein while it's actually being transcribed. And as you can see here, it's also possible for multiple ribosomes to be on one strand of RNA at the same time, coding for, it could be the same protein or it could be many different proteins. So why is this possible in bacteria and not in eukaryotes? So the main reason is that in eukaryotes, this process is separated by the nucleus. So what that means is that DNA will convert to RNA inside of the nucleus, and then that RNA will have to be exported outside of the nucleus into the cytoplasm, and then that's where the ribosomes are, and then those ribosomes can start translating that RNA into protein. So it's kind of like hip hop. So you have a partner, but you're kind of separated. You're not joined together. But in bacteria, there is no nucleus, so transcription and translation can be coupled. So while this RNA polymerase is converting this DNA into RNA, a ribosome can come bind onto that RNA and translate it into protein. So this would be more like ballroom. You have a partner and you joint together closely and doing things in sync. So it's assumed that this transcription and translation coupling applies to all bacteria, but a recently published paper from Johnson suggests that in Bacillus subtilis, this transcription translation coupling isn't actually happening like we think it has been for the last several years. So what they propose is something called runaway transcription. And basically that means that the rate of transcription is much faster than the rate of translation. So the RNA polymerase will actually run away from the ribosome, causing that coupling interaction not to happen because the rates are so different from each other. And the thing about this paper is that they did this with enzyme kinetics and a little bit of sequence data. So they didn't really have any structures to show with this information. So my approach is this transcription translation coupling complex conserved in Bacillus subtilis? And basically, is it more like hip hop or is it more like ballroom? So to do this, what I did was I got the structures that have been solved for Bacillus subtilis that take part in these coupling interactions, and actually recreated this transcription translation coupling complex in a software called PyMol to see if the active sites were actually possible of forming these interactions that we observe in E. coli. So here we have the created transcription translation coupling complex and with the Bacillus subtilis machinery. So the way I did this was I took the structures of the ribosome and RNA polymerase and used some well-conserved sequences in both the Bacillus and E. coli structures of this to align them and superimpose them on each other. And then for the two proteins NES A and NES G, those structures weren't solved in Bacillus subtilis, but we do have the protein sequence. So what I did was took the protein sequence and reconstructed them using homology modeling, which is basically like taking the sequence and using a database of known sequences of similar known structures of similar sequences, and kind of generating a model from those sequences that would best fit the sequence that we have. So here in this like purple bluish color, I have the 50S of the ribosome are the large subunit. In the yellow color, we have the small subunit of the ribosome. In the cyan blue color, we have NUS-A, and then in this red color, we have NUS-G, and then in this green color here, we have RNA polymerase. And the thing about this is, it's kind of a lot. It's kind of hard to see. So what I'm going to do is kind of break it down and take away some of the parts that don't really interact in this in these interactions. So here we have the same structure without the large subunit of the ribosome, and it's still a little hard to see. So what I'm going to do is zoom in on this area right here and just focus on the active sites. So here you can see that these are the active sites of the 30S, the small subunit of the ribosome. In the green, we have the active sites of the RNA polymerase. And then in the cyan blue color, we have NUS-A, and then in this red color, we have NUS-G. And as you can see from the structures, it looks like there are places where these interactions can actually happen and facilitate these coupling interactions that we see in E. coli. But when I actually superimpose the E. coli NUS-A and NUS-G onto the structures as well, we can see that there are some differences in the machinery between the two. So in pink, we have the E. coli NUS-A, and then in white, we have the E. coli NUS-G. And you can see that there are some parts where the structures don't really match up all that well. So for E. coli, we have this large overhang in the NUS-A part. And then in NUS-G, we have this double alpha helix coils that aren't matched up in the Bacillus subtilis structure. So what I'm going to do is actually zoom in into this NUS-G interaction and show you that here in this white part, we can see that there are no matching structures from the Bacillus subtilis. So, and you can see that it looks like there would be a nice interaction between RNA polymerase and the NUS-G right here. So that kind of leads me to believe that maybe the transcription translation coupling complex isn't actually a thing in Bacillus subtilis. And then now what I'm going to do is actually look into this NUS-A structure a little more. And here we can see more clearly where the structures don't align. And what I'm going to do is actually zoom in right here. And then here in orange, we have the E. coli structure for RNA polymerase. And as you can see, that there are some residues that don't line up with the Bacillus subtilis structure. And as you can see that in these new residues from RNA polymerase, there are parts that look like they would match up very well with some of the existing Bacillus subtilis structures in NUS-A, as well as this new structure from the E. coli in NUS-A as well. So as you can see, while it looks like these structures might have places to interact, there are clearly not as much interactions that you would see in the E. coli machinery. So what are we seeing here? Basically, the difference in structures between Bacillus subtilis and E. coli point towards Bacillus subtilis actually using this runaway transcription method, and coupling might not be as viable in Bacillus subtilis as we think it is. And this could be a huge breakthrough for the future, because the way antibiotics work, or at least 2/3 of them, they actually target some of this replication machinery. And if we're suspecting that coupling is going on in these species of bacteria and it's actually not, that means that we're not efficiently targeting these machines as we could be. So future steps are to look more into the actual sequence alignment. So taking the actual protein sequences and aligning them with the insertions and gaps to see how well these residues actually line up. And then once we have that, I can put those aligned sequences onto the structure, and it'll kind of give me like a heat map and it'll show where these structures are, where the residues are conserved in these structures and where they're different. And if they are conserved in the active residues, it could be good to show that this coupling is a thing in Bacillus subtilis. And if not, it will show that maybe this runaway transcription is actually how they undergo the transcription and translation in Bacillus subtilis. And with that, I'd like to acknowledge the Blaha Lab, mainly Dr. Blaha, for helping me through all of this. And I'd also like to thank the Marquis Star Program and the NIH for funding this research. And with that, I open myself up for questions.
Thank you so much for your presentation.